All right, everyone, welcome back to the channel. Today, we're going to be doing questions 14 to 25 on the 2015 USABO Open Exam. So this is the second part of a 4-part series for the 2015 Open. So tomorrow, I'll probably be posting a video for questions 26 to like 37, 38, and then finishing off the exam on Saturday or Sunday. All right, so let's get straight into the questions. First one, question 14. Angiosperms are dominant in most terrestrial ecosystems. Which of the following traits does not contribute to their dominance over the specified clade of terrestrial photosynthetic organisms? All right. Just straight off the bat, reading it, I know that most land plants do have a cuticle, so automatically I can say that A would be the answer. So a cuticle is like that waxy layer on many plants, and it's also present in ferns. So this is not something that's a difference between ferns and angiosperms, and that would make A the correct answer for this question. Even if you don't know the other questions, if you know like what the cuticle is, like what the waxy layers like its purpose is, you can kind of infer that it's pretty ubiquitous among all land plants. So that's why A would be the answer to this question. All right, next question is 15. Which of the following statements about long distance transfer systems in plants are true? Phloem require rigid cell walls to prevent implosion. That is not true. I think this is actually referring to xylem. Phloem have more what's it called? More like flexible cell walls. They're not very rigid. So A is not correct. Phloem transport can be multi-directional. Yes, that is definitely true. If you look at like the sink and sink source and sugar, like that transport system within phloem, I think that's mentioned in like between like chapters 35 and like 39 in Campbell's. This is a correct statement. And then phloem transport is dependent upon metabolic activity. Well, this is of course true, because this phloem transport requires ATP, and you're gonna get that ATP from like the metabolic activity. So C has to be true. So clearly, I know this might not just be a one answer question. Maybe I'm gonna have to keep that in the back of my mind that these questions might have more than one answer. The driving force that moves water in the xylem is ultimately derived from the energy of the sun. Yeah, because the energy of the sun is what drives transpiration, and like it can cause water to like evaporate. So let's say you got a plant here. I'm just gonna like draw a plant, and then you got the leaves. Let's say you absorb and whoops, let me just draw some roots here too. Like let's say you absorb some water. The sun is gonna evaporate the water. Oh, that's not a good sun. Let me draw a little bit like this. All right. The sun is gonna take away this water right here that's present up here, and then through capillary action and everything, the water is gonna flow up. So if the energy of the sun is gonna determine how the water is gonna move. So that's why this would be correct. So D is correct. And then xylem transport occurs through the living xylem conduits. That is incorrect. Xylem cells are dead, yet they still function. So here, the answers would be B, C, and D. All right. Number 16. You are given an Arabidopsis mutant PIN3 in which an auxin efflux protein is disabled. So clearly I need to remember what auxin does. It's a very important plant hormone, one of I think 5 or 6 plant hormones you need to know very like down pat. And then which of the following phenotypes would you expect? All right, so definitely not A. It would not be accelerated seed germination because auxin is being disabled. Yet this one does seem pretty correct. Delayed seed germination. Diminished phototropic response, that is obviously correct. That is the main function of auxin. Like, if you search up what does auxin do, that is the number one thing that comes up. So that is definitely one of the answers. And increased rate of fruit ripening, that is not correct. I think that is ethylene gas. And then reduce stomatal response to soil drying. That's not correct either. Now, here's where the tricky part comes in. So clearly the answer seems to be B and C, but the answer key only has one option that you can pick. So, and they say the answer is C. I don't know why it's not B. Maybe C just seems to be the best answer, but yeah, the answer key says that it's only C, but based off like what auxin does, it should be B and C. So just keep it in the back of your mind. The answer to this question is C, but the key is saying it's B and C. But yeah, knowing auxin and its functions, as well as like ethylene, abscisic acid, what else am I forgetting? Oh, it's just on the roof of my mouth. There's a couple of more plant hormones that you really should know for the USABO, because they're really, really important. And then we got number 17. So Chrissy is a papillon. I don't know what a papillon is, but I don't think it really matters. Papillon, okay, it's a herbivore. So I'll just underline herbivore, who tends to round out her daily serving of crunchies with slash palms and grasses found in the yards. Today she decided to try a new plant, which I have no idea how to pronounce, like Dieffenbachia. In the living room, unfortunately, she developed the following symptoms: swollen lip and a mouth accompanied by drooling. Which of the statement best describes Chrissy's reactions to Dieffenbachia? And her symptoms were a result of the following effects. All right, so trichomes could be possible, but I'm very, very doubtful. Actually, the way I would solve this question is, I think in chapter 39, which talks a lot about plant hormones, you can go to the very end of the chapter, and it's herbivore defense against plants defense against herbivores. And one of the things that it mentions is that how plants can sometimes produce an amino acid that can cause these certain responses in the herbivore. So that's one of the things that can happen. And the other option isn't really, like, the other mechanism isn't really mentioned here, so I won't really touch it. But yeah, so because plants, they can produce compounds to, steer away their predators, which in this case are like herbivores. This is not a tissue level, not an organ level, but in fact, it's a cellular level defense. They're going to produce a compound which could be like an amino acid. It could be a certain chemical, which they release which they release, and then the herbivore takes that in, which affects them. And that is definitely a cellular level defense. It's not something that happens at an organ level. It's a cellular level defense. So you can kind of infer that, but this one was a little bit different. A little bit difficult. You don't really need to know what the specific plant does, but kind of need to know the general idea of how plants defend against herbivores and other organisms. All right, 18. In which of the following habitats would you expect to find many C4 plants? So C4 plants, just to recap, are a way that plants can adapt to hot environments. So, normally what happens is you have the light dependent reaction, and then you have carbon dioxide undergoes the Calvin cycle, and and glucose and everything is produced. So G3P, sorry, and there's like so many, all this like compounds and everything. But here with C4 plants, what happens instead is that you have PEP carboxylase, which is an enzyme that converts PEP into a 4-carbon sugar, which could be like oxaloacetate, and then malate, and then it makes carbon dioxide. So it's kind of like a longer process, which is actually thought to be more efficient. But yeah, this is for hot and arid. Actually, I don't know about arid, but I know for sure this is hot. This might not actually be arid environments, but this is definitely hot environments. So cool temperate grasslands, that's not hot. Subtropical grasslands and tropical rainforest canopy, that is definitely one of them. Subtropical grasslands and tropical rainforest understory. So the understory doesn't really receive a lot of sunlight. And sunlight is a really big part of C for plants. So that's why C wouldn't be an answer. And then temperate forest canopy. Well, temperate forest isn't as hot as a tropical rainforest, so D wouldn't be correct. And then the understory, as I mentioned, wouldn't be correct. So yeah. The answer is then B. So yeah, this is mentioned, I think, in my textbook. It was in chapter 10, photosynthesis, and it talks about C3, C4, and then CAM plants, which I'm not touching on, but are very important to know as well. Okay. So the answer to number 18 would be B. Number 19. All right. You hypothesize that reduced caloric intake may increase the lifespan of C. elegans. To test your hypothesis, you feed the worms different caloric diets and measure their lifespan. All right. Which of the following are examples? So, of a positive control experiment you could perform to further support your result. All right. First option, A, knock out a gene that decreases the worm's ability to eat and provide the strain of worms with the same dietary regimes. This is correct, I believe, because, a positive control experiment, like in simple terms, is kind of like an experiment that a scientist performs to kind of like validate already given results. So this will determine how like if you're decreasing its ability to eat, that could like affect its lifespan and such, because we're looking at reduced caloric intake. That's kind of like what we're studying, and decreasing its ability to eat would definitely reduce caloric intake. So this could like, kind of prove whatever the scientist is trying to look at. Provide one group of worms with a drug known to increase their lifespan. I guess this would be correct because we're also looking at like how the caloric intake is increasing their lifespan. So I think one group of worms, that's like if it's increasing their lifespan, that could kind of relate the caloric intake and lifespan. So I would say that is correct. Provide one group of worms with their normal diet and measure the difference between control and treatment groups. Well, if you're giving them their normal diet, that's just a control group. That's not really a positive control experiment, and you're not reducing caloric intake or anything like that. So you're not really testing your variable, which is what a positive control experiment is all about. So C would be incorrect. Provide the worms nothing but sugar, water, and measure the placebo effect. I don't think you need to measure the placebo effect when you're looking at a positive control experiment. You're just looking at whether, like, reaffirming those results. So sorry, D doesn't seem correct either, because we're not looking at that placebo effect. These two right here, C and D, would be more reasonable if we were like originally testing our hypothesis, not doing like a positive control experiment. So this would be like one of the first few experiments that we do along with our original experiment. And then E, isolate one group of worms known to have longer lifespans determined by genetics and provide them with the treatment diet. Yeah, so this could look at, they have a longer lifespan, and then you could look at their genetics to see if that's because of reduced caloric intake. So further analysis could show that. So that's why E could be an answer. So therefore the answer is A, B, and E. So a little bit of a tricky question, but really plays on the importance of knowing like the scientific method and such. Yeah. Let's go to number 20. So you are a summer student At the Marine Biological Laboratory Woodholes, like Hodgkin and Huxley, you want to study the speed of action potential propagation in the squid giant axon and compare it with a cultured neuron isolated from the central nervous system of a mouse. To do this, you apply a stimulus near the squid and the mouse axon hillocks and record the speed of the action potential propagation downstream of each. You calculate the squid and mouse speeds to be 25 meters per second and 145 meters per second, respectively. Based on your observation, which of the following is true regarding the properties of your test neurons? All right. Right off the bat, we know that the speeds are different, and we know that the size of the neuron is different. So that could be a very important thing because when you have a wider axon, that could mean that there's less resistance, and that could increase the speed of the action potential. So that's something just to maybe keep in the back of your mind. All right, so which of the following is true? The squid giant sorry. The squid giant axon is myelinated, and hence the propagation speed is high, even though it is an invertebrate. Well, that's clearly incorrect. Its speed is quite low. It's 25 meters per second, so one is not correct. The cultured mouse axon is myelinated. That's quite probable because of how fast the action potential is going through the axon. And then, therefore, the speed is greater because there are voltage-gated ion channels throughout the myelination. Well, yes, there are gated ion channels, but that's not necessarily I think they both have the voltage gated ion channels. Myelination is just like meaning that they have more of those voltage gated ion channels. So I think that's a little bit vague. I'm not 100% sure, but I'll just put a question mark next to it. The squid axon has a larger diameter, which reduces the resistance. This is what I mentioned before, thereby increasing the speed of propagation. Well, yes, even though the speed was much smaller, this logic is completely true. The only reason, or I don't know if this is the only reason, but the major reason why this squid had a slower action potential was because there had to be continuous conduction because there was no myelination. So that's why C3 would be correct. And then the cultured mouse axon is myelinated. That's true. And therefore, there's a solitary propagation of action potential. That is 100% true. So, okay, well, even E is bolded, but yeah, E is the one that makes the most sense. Here, the key was knowing that what is saltatory and continuous propagation. So just like a quick recap, you have your soma, with their dendrites here, and then you have the axon, and then the axon terminals and everything. This is a terrible drawing, but it kind of gets the job done. When you have this these myelin plaques, this is allowing for voltage gated ion channels at each of these nodes, which is each node is speeding up the reaction. So if you just have no myelin, that's not going to speed up the reaction at all, because you can't kind of rejuvenate that voltage gated ion channel effect. So that's why 3 and 4 are correct. 21. You are a developmental biology lab. You are in a developmental biology lab, and your professor has asked you to study the effects of manipulating cortical rotations on a fertilized frog embryo. All right. I'm not very experienced with this type of stuff. So if you do know more about this, please write it in the comments or let me know. Because I'm not really good with animal development stuff, but let's see. You try a few manipulations and report to your professor which of the following. Okay, so this is only one. Which of the following is an incorrect observation? Well, based on what I'm seeing, I know for sure the answer is going to be either A or D. Because both of them, you're inhibiting cortical rotation, but they're leading to different results, which is not possible. It's either going to be ventralized or dorsalized. And in this case, it is just going to be ventralized. It's something I remember. I'm not sure about the other options. It's definitely not going to be a normal embryo. And I don't know about conjoined twins, but I know for sure it's going to be A or D because, of that. They have like the same reasoning, but different responses. So, that's going to be one of the answers. And I'm pretty sure that inhibiting cortical rotation will lead to ventralized embryos. So therefore, this would be a correct answer. Sorry, a correct response, and D would be the correct answer. So this is a valid statement. Like, this is true, but we need to find what's false, and this is the one that's false. So this would be the answer. But yeah, if you know more about cortical rotations, please let me know. Always willing to learn more about this stuff. All right, number 22. You are making a fate map of a developing Xenopus laevis embryo. Which of the following structures will the archenteron form in the adult animal? All right, so this one is actually quite an easy question because if you know what the archenteron is, this just becomes so much easier. The archenteron is kind of like a precursor to like the gastrointestinal system. So because the lumen of the gut, which is something I remember from cows, but like the lumen of the gut is definitely part of the gastrointestinal system, so that is going to be the correct answer. All of these other ones like blood vessels, epidermis, they're not part of that system, so they're not going to be the correct answer. So that's why the answer would be E. All right, reptiles differ from amphibians due to A, eyelids and external ear openings. Well, the eyelids thing just isn't something that would make them different, I don't think. And then external ear openings. So I know for a fact that reptiles do not yeah, I'm not really sure about the external ear openings things, but this is definitely not a similarity. I'm sorry, this is not a difference. I feel like both of them do have eyelids, so this is not an answer. Scaly skin. Oh yeah, that's kind of given with reptiles. They're much more scaly. Amphibians are much softer because they live in both water and land. And then a cladoic. I don't think that's how you spell it. I think there's an I between the E and the D, but this is the correct answer. Of course, everyone knows that snakes, lizards, they all have scaly skin. A cladoic egg is like an egg that is surrounded by like a hard shell. I think it's usually calcium carbonate, and reptiles definitely have that, but that is not present in amphibians. So I will mark that, but I'll just read the other options just to see. Terrestrial lifestyle and the ability to climb. That's not really something that would be a correct answer, because reptiles and amphibians technically both live in terrestrial environments. Amphibians do spend some time in the water, but they can also live on land. Scaly skin, so that we know is correct. And then an external ear opening. Well, I was kind of unsure about the external ear opening, but I don't think that would be an answer. Because I know for sure that scaly skin and cladoic eggs are something that only reptiles have and not amphibians. And then a three-chambered heart and cladoic egg, no. That's not a difference between the two organisms. I know that reptiles and I think amphibians both have three-chambered hearts. So yeah, the answer just seems to be B. Number 24. While there are several types of reproduction in a single-sex amphibian species, blank is characterized by offspring that have the exact genome as its mother. This is also true in blank, except that this requires the presence of male gametes. All right, even if you don't know what this is, you can kind of eliminate numerous options. I know, so parthenogenesis is this one. It's related to just the mother, and then gynogenesis is related to just the father, or requires the presence of the male gamete, so that's why the answer would be A. But neogenesis and hybridogenesis seem to be the other answers, and they're not correct because neogenesis, I think, is the Oh, what's neogenesis? I think it's like the making of new tissue, I believe. And then hybridogenesis is like some weird type of reproduction where I think it's like the reproduction of like two hybrids or something. But yeah, parthenogenesis and gynogenesis are what make the most sense. Or gynogenesis, maybe. I think I'm pronouncing that incorrectly because that kind of looks like gynecologist, but you know. But yeah, this was just like a trick of the Latin roots, if you remember them. So many people know what genesis means, but partheno and gyno are what like the key terms that you should know. Or so yeah, the answer to 24 would be A. And the last question for today, which of the following structures of the human nephron is relatively impermeable to water? All right. Ascending limb of Henle, descending limb of Henle, glomerulus, medullary collecting duct, and proximal tubule. So yeah. The answer to this question seems to be not B, because what I remember is that, like for the loop of Henle, this is actually not a very accurate drawing because it's supposed to get thinner, but let's just say this is like the loop of Henle. The ascending limb is impermeable to water and permeable to salts, and the descending limb is permeable to water but impermeable to salts. So I know A is correct. And then glomerulus, we have some like filtration stuff going on in there, so it can't be impermeable to water. And the medullary collecting duct, of course it can't be, because a collecting duct, there definitely needs to be water transfer that occurs. I think that the ascending loop of Henle is the only one in this option that makes, in these like option choices, that makes sense. And the proximal tubule, of course, cannot be an answer. So yeah, that's why the answer to number 25 would be A. So this is just kind of like touching on renal physiology. And I think on the open exam, they should tell you whether an option has more than one answer. I don't know. On the online one, you could kind of tell because it let the computer either let you select one answer or more than one answer. But yeah, I don't think B, C, D, or E make any sense. So the answer seems to be A. All right, everyone. That is the end of this video. So tomorrow, as I mentioned before, I'm going to be posting questions 26 to maybe like 37 or 38, and then posting the rest of the exam on Saturday or maybe Sunday. I hope you guys enjoyed this video. Please let me know if you have any questions in the comments below, or if you have anything to add to any of the content, or like, make my explanations more like deeper, or any like comments like that. Please write in the comments below. Thank you guys so much for watching, and I'll see you next time.